how are you doing? Good, glad to hear. So today we're just going to go over some protein synthesis, so how we get our proteins from our DNA, because we know that DNA is the coding block for proteins, tells our body what proteins to make. So we're going to go over how we get from the DNA in the nucleus to the proteins that our cell makes. So that is two processes, transcription and translation, and I'll explain both of them shortly. First, I'll just share my screen. Alrighty, so let's come over here. We have a nice long strand of DNA. So along here, we've got our phosphate sugar backbone. So that's kind of like our spine of our DNA, and then all our bases coming along here. Now, normally we would have another strand parallel to this, but I've just drawn one for simplicity. So our bases are what we call the monomers of our DNA, or they're part of the monomers, sorry, of our DNA. Can you tell me what our monomer is for proteins? So what is the basic building block for protein? Yeah, amino acids, very good. So that is, in order to get from DNA to proteins, we first need to get from DNA to amino acids. And one of the ways our body likes to do this, or the way, is each of these, so DNA reads itself in threes, in triplets. So every three bases, so for example, TGA is going to be a triplet, and that is what is going to code for an amino acid. So each three bases, each three different of the letters is going to code for an amino acid. So for example, this TGA here would code for an amino acid, and then if we had CGT, that would code for an entirely different one. And something important to remember with this, that I think you have already covered, is that these are discrete. So you read our mRNA, which I will cover in a second, is going to read TGA, and then it will have a new example or new triplet, CGT. It's not going to go and overlap. It won't go AGC. But this will make more sense later, so I will explain and elaborate more in a second. But the first thing we need to do, we'll come over to our pretty little cell here. So in order to get from DNA to protein, we need to change locations within the cell. So remind me again, where is our DNA within the cell? Yeah, the nucleus, well done. And where do we make protein? Not sure? Yeah, okay, fair enough. So we make proteins in these little purple dots that I've just put a few in. There's normally a lot in cells. Little purple dots here, and these are called ribosomes. So they are scattered throughout the cytoplasm. There's lots and lots of them, and they're super duper tiny. They can be free in our cytoplasm, or they can be part of our endoplasmic reticulum. Do you remember endoplasmic reticulum? Yeah, perfect. So these little guys are responsible for protein synthesis, but our building blocks, or not our building blocks, sorry, our templates for our proteins are all inside the nucleus. And we know that DNA isn't going to leave. It's not going to wander around the cell. So how do we get from our DNA in here out to the ribosomes? So that is the first issue that we deal with, and that is solved by the process of transcription. So what happens? We'll zoom in here. So here we have a nice piece of double-stranded DNA inside our nucleus. Now what's happened is we have this enzyme here. So this, let's call that RNA polymerase. So this enzyme is going to bind to a section of DNA that we call the promoter sequence. So this promoter sequence is at just a section of DNA. So it's just a section of DNA plus a few proteins alongside it. And it basically is just telling the RNA polymerase, hey, gene starts here if you want to make this protein. So the RNA polymerase is going to bind to this site. And then the next thing it's going to do is unwind the DNA and separate our 2 strands. So this is a similar process to, do you remember doing DNA replication where you have to split the strands? Yeah, so it's very similar to that. So we have to split the strands, and then we're going to use one as a template. So yeah, same principles as DNA replication. So let's go from here. So we have our nice little enzyme, and then it untwists and breaks all our bonds here. Now I'm going to say that we have our bottom strand here. This is going to be the template strand. So if this is our template strand, and we're making some RNA that's going to look, what's the RNA going to look like? Yeah, correct. It's going to look like this DNA strand because it is complementary bases. So yeah, just a quick note on that. RNA is very, very, very similar in structure to DNA, but instead of having your 2 strands, it's just got 1, so it's smaller as well. And then it has, instead of thymine, so T as the base, it's going to have uracil, so U. And the reason it's different names, DNA and RNA, so DNA is deoxyribonucleic acid, RNA is ribonucleic acid. So RNA just has 1 more oxygen than DNA, but it's just a slightly different structure in the sugar. But the main point is that it's smaller and it's single-stranded. So we're trying to make a smaller single-stranded thing. So if we have this as our template strand, so we'll go back down here. Here's my nice little template. And here's my RNA polymerase again. So this RNA polymerase is going to keep going. It actually does the whole thing. So the RNA polymerase is here on my promoter. And it's basically just going to travel across like it does, like DNA polymerase does in DNA replication. So travel across and add bases that are complementary with our bases here. So it's copying off of, it's going to be a copy pretty much of this strand here. So, and these have names. So our template strand is also known as the antisense strand. And then the strand that the mRNA is going to look like is what we call our sense strand. That's our sense strand there. So what we end up with is we create this nice long strand of our RNA, which I've got in pink just because it's a different sugar. But this is called mRNA, so it's messenger RNA. So this is a messenger. And just a note as well, you can see I've got a slightly different green for these bases. That's just to indicate that this is uracil instead of thymine. So it's the U instead of the T. And another important note is that our little RNA polymerase here works from the 5' end to the 3' end, and that is on the sense strand. So what I mean by that is this end here is going to be 5', and this end here is going to be 3'. Do you understand the difference between 5' and 3'? Not really? Okay, that's all good. This is a little bit, I'll draw it down here. It might be a bit more depth than you need to know, but I will explain it just so that you understand. So let's draw ourselves a nice little DNA molecule. So this is just a nucleotide. So this is our monomer for our DNA. So here we've got a phosphate group, hence the little P. And then here is where we have our bases. So there's your B. And then down here we have an OH, so a hydroxyl group. So this phosphate group is what we call 5' because it's on the 5th carbon. So there's a carbon up here that's the 5th carbon on this sugar. And down here is the 3rd carbon for the hydroxyl, so that's called 3'. So when we line these out, so let's say I rotate this guy, we have another one, and then rotate him. More and more. They rotate, so say we're doing that, and then this will join on, because we've got our phosphodiester bonds, you'll form a little bond here. So you'll form a bond between the 5' and the 3'. And this will keep going and keep going and keep going. But eventually at one of the ends is going to have just a 3', so a hydroxyl group hanging out by itself over here. and one of the ends is just going to have a phosphate hanging out by itself over the other end. So that's why we call them 3' and 5'. I don't think you're going to need to know about which carbons things are on, but it's helpful to understand that 5' is where you've got a loose phosphate, and 3' is where you've got a loose hydroxy group. And they're always opposite. Like if you have a DNA strand going this way, so it's got 5' on this end and 3' going this end, then the one going the opposite direction is going to have 3' here and 5' here, because DNA strands do run in opposite directions. Does that kind of make more sense? Yeah, okay, perfect. So yeah, don't stress about the perfect structure of these. Just remember that we've got our phosphate and our hydroxyl groups join, and on either end we're going to have a loose end, and that's what makes it either 5' or 3'. Perfect. Okay, so coming back to here, where we've got our nice new RNA molecule. Can you remind me, what's this called again? mRNA. Perfect. And the reason we call it mRNA is because what happens next is this RNA polymerase reaches a sequence of the DNA on our template strand called the termination sequence. So this basically tells the RNA polymerase that the section of DNA to make the protein is done. So the RNA polymerase will go, okay, it's my time to go away, and it will detach itself as well as the mRNA. So both of those will detach and the DNA will reseal. So the hydrogen bonds between the bases will reform and retwist up. So you've got now, you have a nice piece of RNA that is just floating free in the nucleus, and it's small, so it can go places, because in our nucleus, we've got our nuclear envelope on the outside. Do you remember that? Yep. But the nuclear envelope isn't fully solid, it's got pores. So if we have a little, let me match the color. If we've got our little pink mRNA in here, it may not be, it's not as big as if we have a big old DNA guy like that. It's a bad example. It's not true to scale, of course, but this little mRNA is a lot more able to leave the nucleus than our big DNA molecule. So that's exactly what it's going to do. It's going to leave the nucleus. Although one thing it does do before it leaves the nucleus, so we've made our mRNA and it's free, but some enzymes are just going to get rid of any introns within the mRNA. Do you know what introns are? No, that's all good. So an intron is a portion of DNA or genome that doesn't code for a protein. So for example, that would be your promoter and termination sequences. They don't actually code for proteins. They're just there to help the enzymes bind to the DNA. So we want to get rid of those because there's no point in all of those little useless bits of RNA getting out and into the cytoplasm. We don't want that. So enzymes will just take out the introns. So I always remember those as introns because they stay in the nucleus. And so everything that is left in this RNA is going to be called an exon. So that is stuff that exits the nucleus. And then the RNA is free to float around. Ooh, that's too big. It's going to come and float around in our cytoplasm. So that whole process of how we make our mRNA, so messenger RNA, is called transcription. Is that making sense? Yeah, awesome. Please feel free to ask me questions at any point. Okay, so moving on, what happens next is we have our RNA. So this is our nice pretty little RNA here, and it comes across our ribosome. So this is our ribosome in purple. Now our ribosomes are made of two subunits, so it's called, really nice and easy, it's called the big subunit, or the large subunit, and the small subunit. So it's fairly easy to identify. And these are both also made out of RNA. So this type of RNA is called rRNA. I'll do it in the purple to match. This is rRNA for the ribosome. And that is what is made in our nucleolus, but we'll cover that later. So these two units made of rRNA, and then we have our mRNA, which comes in and it binds to our small subunit. So RNA comes in from the nucleus and binds to a small subunit of our ribosome. Now, what happens is within our cytoplasm, so just freely floating throughout the cell, we have some little molecules called tRNA, so transport RNA. So that is basically, it's just a little thing like this, and it has an amino acid. It always will carry an amino acid, like so. So there's your amino acid. And then it will have binding sites for the bases on RNA. So it'll have some little binding sites down here and be able to bind to our RNA. And these will be individual. So these will correspond to the amino acid. So you can't really have a combo between what amino acid you have and what bases or what chemicals you have that bind to the bases down the bottom. So it means that every amino acid is going to correspond to our bases. Does that make sense? Cool. So the first thing that is going to happen is a start codon will be the first part of the RNA. So remember how I was talking about triplets with the DNA, how we have 3 pairs are called a triplet? So 3 pairs of DNA are called, 3 nucleotides of DNA are called a triplet. 3 nucleotides of RNA are called a codon. So that's just, if you hear me talking about codons versus triplets, that's the main difference. Triplets is DNA, codons is RNA. So we have what we call a start codon. So that is a 3 base pair sequence, so a codon, and it is very specific. It has to be AUG. So adenine, uracil, and then guanine. And that tells the tRNA, or the tRNA will have a chemical on it that signals the start of a protein synthesis. So the tRNA that will be able to form bonds with the first start codon, so AUG, is going to have a little chemical on it that tells us to start. So, does that make sense? Yes, perfect. So, what happens is our tRNA molecules, they come in to our large subunit and bind to their complementary codons. So, they will be able to form hydrogen bonds. It's a similar process to how our DNA forms hydrogen bonds with each other, so it is complementary. So they'll form hydrogen bonds with 3 of our base pairs. So a codon will form hydrogen bonds here, and then this amino acid, or in this case, the start chemical. So here and this happens 2 at a time. So there will be 2 tRNA molecules, and they come in and we have a start codon, and then this one is just an amino acid. And so what happens is, so once this start codon is happy, we've started, what will happen is the tRNA molecule is going to leave. So this one's going to leave, and then this RNA is actually going to move over. So I think of it as like a conveyor belt. It'll just move over 3 spots. So this tRNA molecule will stay, but it will move over to this spot. So it's moving up the queue, and then a new tRNA molecule will come in. So I've got a bit of a diagram of that here. So we've got, this is just moving along kind of 3 at a time, and a new tRNA molecule is coming in. And what happens is it forms its complementary bonds with our bases here, and then each of these is going to have an amino acid on it. Yeah? So then the ribosome allows there to be these bonds formed between amino acids. Can you remember what the bonds between amino acids are called? Yeah, peptide bonds. Well done. So peptide bonds are forming between these amino acids, and that is allowed to happen by the ribosome. So the ribosome is like, normally you'd have an enzyme for that. The ribosome is the enzyme, basically. So that is allowing those bonds to form. And once the tRNA detaches itself and moves on and allows the next one to come in, like the conveyor belt, these amino acids stay. So you can see here we have the amino acids of the previous tRNA molecules. So the tRNA molecules go back into the cytoplasm without their amino acids. The amino acids stay here and form our nice chain of proteins. Does that make sense? Yeah, great. Okay. And so how we get to the end is similar to how we start. We have stop codons. So we actually have 3 stop codons. These are UAA, UAG, or UGA. So either one of those 3, let's say this is UAA here. So once, for example, let's say that we are all the way at this end here. So this end here is up here. We have one last tRNA with an amino acid here. And then this here is going to be our last tRNA coming in. This RNA, this tRNA, is going to have the stop signal on it. So basically it'll just say stop, and then no more peptide bonds are going to happen. Our protein is going to release from the tRNA, and it's going to go off and be processed. And our mRNA is just going to unbind and go and degrade within the cytoplasm. Cool. Okay, so that is basically our process of how we make proteins from DNA. Is that all making sense to you so far? Yep. Do you think, can you sum up to me the 2 things that need to happen in order for us to get there? Oh, and sorry, this whole process of getting from our mRNA towards our tRNA, towards our amino acids is what I call translation, or what we call translation. And a way that I like to remember the difference, because, you know, they're 2 T names, it can be a little bit tricky, is translation is when we go from one language to another. So in this case, it's where we're going from nucleotide bases to amino acids. So it's like 2 different languages. Whereas transcription is when you write something down in the same language. So that's what we've got up here. So we're going nucleotide bases, nucleotide bases. So same language, transcription, different language, translation. Okay, perfect. So, sorry, as I said before, what are the 2 things that need to happen? Yep, so DNA needs to find a way to get out of the nucleus, or in this case, it's not the DNA itself, it's the mRNA. Yeah, very good. And then we need the mRNA to be translated into protein. Perfect. Okay, so I can send you these diagrams if you like, and I can annotate them a bit more. But that is basically how we form proteins. Yep, so after we have this formation, it's basically going to go into our endoplasmic reticulum. Remember how you said you like that? Yep. Going to go into our endoplasmic reticulum, which these ribosomes are often attached to, and it's just going to fold itself up and be processed and ready to send out into the body for its functions. But we can talk about that one later, because that is, the folding of proteins is a bit of a different process. Okay. Amazing, so great work today. And do you have any more questions for me? No? Perfect. Feel free to message me if you do have any questions throughout the week so that I can think about them and then we can address them in our next session. And yeah, otherwise I hope you have a great week and I'll see you next time. Thank you.